Hello, everybody. I'm Camille Rustenholz, and I'm an associate professor at Strasbourg University in France, and I'm performing my research in grapevine genomics at INRAE Colmar. And today I am in charge of recording the online lecture that is going to prepare you for manual curation of PN40024 and this is going to be in the frame of the Annot Training School that you're going to follow next week, and which is part of the INTEGRAPE cost action. So first, I'd like to tell you about what is annotation so that we are all on the same page before starting this training school. I'd like to start with two definitions and to differentiate between structural annotation and functional annotation. So structural annotation is about locating the genes and coding regions in a genome assembly. And these genes or coding regions, they should have a correct structure. So which means having a start codon, a stop codon, and a canonical exon intron borders. We're going back to that later on. So this is structural annotation. And we'd like to differentiate that with functional annotation. So functional annotation is actually to identify or predict the biological function of the genes. So let me make here an analogy between structural annotation of a genome and locating the words in a book. So here you have a book where you can find some words. So for example, hallo, wie geht es dir heute? And on the right, you have a DNA sequence, and the challenge with structural annotation is to actually locate the words, locate the genes that are encoded in this DNA sequence. So it's not the exact same problem by locating the words in a book comparing to locating genes in DNA sequence, because in a DNA sequence, you have many letters or nucleotides. And so it would be just as if you have a book with many letters like that, and you have to find the good words. So for example, here the words are here, hallo, wie geht es dir heute? But again, with DNA, it gets more complicated because you can have words that are forward, like wie, or backwards, like hallo here. So we have to locate and find words in a book that can be forward and backwards. So this is the same challenge as locating genes in a DNA sequence. So this is the DNA sequence that we want to structurally annotate. And we can find here two genes. The one below is a forward gene. It starts with an ATG and stops with a TAA, a stop codon, and a backwards gene, which starts with the reverse complementary sequence, here TAC, so ATG, if we translate, reverse translate, and stop with an ATT, or reverse translate TAA. So this is the challenge of performing structural annotation, is locating the genes in a DNA sequence, just as if we would have a very complicated books with a lot of different letters, and we could get words that are forwards or backwards. Then if we want to make a difference between structural annotation and functional annotation, so functional annotation would be to get the meaning of the words, so to understand the words. So for example, with my example here, hallo, wie geht es dir heute? So some of you may understand, I know, but if we want to get the meaning in English, we need to translate it. and it will be hello, how are you today? So this is the same if you are looking for a functional annotation of a gene. We would like to find the meaning, or actually the function of the gene, or the function of the protein that is encoded by this gene. So for example, here, this gene is a fragment of apetala-1 involved in floral identity. So this is a quick example to distinguish between what structural annotation and functional annotation are. So what we are going to perform together is mostly focused on structural annotation. So I'd like to make things very clear with you of what is a correct structural gene annotation, what we also call a canonical structural gene annotation. So you have here a scheme of a gene. So you can find here 5 different exons, and they are split with 4 introns. Everything starts at the 5' end with the transcription start site. And the start codon may occur later, which defines between the transcription start site and the start codon the UTR, so the untranslated region here, which is a 5' untranslated region. Then at the start codon starts the coding sequence, what we call the CDS as well, which is in purple here. And it stops with a stop codon here. And you may have at the 3' end also a UTR, so an untranslated region. And so both UTRs at the 5' and the 3', they may also be split with the introns. About introns, most of the case for introns, they start with a GT and end with an AG. This is for about 95% of the cases, but we can also find in about 5% of the cases, not a T here, but a C. So the border would be GC AG. So to summarize, canonical gene structure may start with a 5' UTR, which is not mandatory. And we will see it's not always easy to retrieve the UTRs when we are performing manual curation. What is absolutely mandatory is that the genes has a start codon, so an ATG codon. The canonical exon intron borders, as I said, are GT AG in most of the cases, and rarely GC-AG in about 5% of the cases. There may be some other options, but which are very, very rare. What I'd like to notice here is that genes may also be a single or mono exon. So this is possible. So you may not have to annotate introns. So somehow introns are not mandatory. What is very mandatory as well is that you need a stop codon, so one of these 3, TAA, TGA, or TAG. And at the end, but it's not mandatory, as I said, for the UTR, you may find a 3' UTR. So this is the structure that we are going to be looking for while performing manual curation. So this is very basic stuff, but it's only to remind you and to everybody so that everybody is on the same page. And what's the difference is that when we are going to perform manual curation, we are not only going to stick with the canonical gene structure I've just described, because you can find structures that look like genes pretty much everywhere in the genome with open reading frame. But what will make our gene, let's say, real or more real, if it has a biological evidence and or has some homology with another gene. And this is really mandatory when we are curating gene. We not only expect the gene to have a correct structure, but also to have some biological evidence or homology, which testifies that it's probably a real gene that we are curating. So if you go back to the definition, so as I told you, we are mostly going to focus on structural annotation next week altogether, less on functional annotation, and we will be performing manual curation, which is going to be about validation of the structural annotation and check if it fits with biological evidences and or if it has some homologous genes. So why do we need to perform manual curation? So there are many reasons for us to perform manual curation. And I'm going to detail all of these. And so I'm going to start with the specificities of the PN40024 V4 genome assembly. And you'll see that it makes manual curation very important. So what you need to know, so this is going not to be very detailed, but just to give you a hint on how we performed this new version assembly. So the idea was to keep as much as possible the high quality Sanger assembly that was obtained in the 12x version. But we wanted to improve its completeness and the contiguity of the scaffolds and the context. So what we had in our hands were the Sanger PN12x scaffolds. So here in red, you can see that there are still some gaps, like ends in the sequence. We also we performed some Pacific Biosciences sequencing, and so we retrieved 27x PacBio reads. So this was the old technology, which had a high error rate of about 15%. So not very accurate reads, but we'll see how we handle that. And we also had in our hands 15x depth of Illumina reads from PN40024 genotype. So altogether, with all this data, we managed to improve the genome assembly. So first, to make long story short, we used the PacBio reads here in green. We performed self-correction using the Canu software. We also used the Illumina reads and the LORDEC software to correct the PacBio reads. So after this first step, we had some corrected PacBio reads to play with. Then we wanted to identify the missing sequences from the previous 12x version. So we aligned the green PacBio reads on the PN12x scaffolds here in red, and all the reads with less than 80% identity and coverage were considered as missing sequence, so part of these gaps represented here. These reads that fell in the missing sequence part, they were assembled individually using the FLY software. Then we merged the PN12X scaffolds with these new sequences that were assembled using Haplomerger 2, and we performed some scaffolding with the corrected PacBio reads and the software Opera LG. So at the end, we were able to retrieve longer scaffolds, which were a mosaic of the Sanger scaffolds, so here in red, along with some dark green sequences, which are new PacBio assembled sequences. But we knew that the PacBio sequences were still, they still have errors, so we needed to perform some polishing and improvement on that. So first, what we did is a gap filling step. So we used the Illumina reads here in blue and the software Gap Closer as the first round of gap filling, so to help resolving the N regions, the gaps previously identified. And then we performed the second round with the LR Gap Closer, which uses corrected PacBio reads. Then after the gap filling, so resolving the N gaps region, we perform polishing of the sequencing. So polishing is correcting the last error that remains using Illumina reads and the software Pylon. So here, what is really important to remember is that this assembly is not a totally de novo assembly, but it's rather a mosaic of Sanger region along with new PacBio regions, and this is going to have some consequences on how we perform manual curation later on. The last step is using these new scaffolds and very dense genetic maps. We were able to anchor the scaffolds to the chromosomes and build what we call pseudomolecules, which are the models for the 19 PN40024 chromosomes. And so with this, we ended up with the PN40024.V4 assembly. So this is the genome you are going to play with during the training school. So I don't want to give you too much of detail, but just to give you to show you that this new version is improved compared to the previous one. There are obviously less scaffolds because this was the whole point of doing this. So getting a longer and more complete scaffolds than the previous one. So we managed to end up with this. The N50 is twice longer than the previous one. So this means that the completeness and the contiguity of the sequence is improved. And what's also noticeable is that we greatly decreased the number of N's, so unresolved sequence, so which were about 15 megabase in the previous scaffolds, and now it's only 1.8 megabase with the new scaffolds. So overall, if we compare the two, so I just explained about the N, so I'm not going back to that. But also, if we consider the chromosome unknown, so chromosome unknown is not a real chromosome, but it's the chromosome that were artificially built by concatenating the sequence that could not be anchored to the chromosome. So in the 12xv2 version, the chromosome unknown was about 27 megabase long, which made it the fourth largest chromosome, so to say. And now it's only 12.5 megabase long. So it means that we were able to anchor much more sequences on the chromosomes. Also, we knew that with the PN12xv2 version, the heterozygous regions were not very well resolved, and this is improved. And we are going to show you that later on. And also, we knew that in the 12xv2 version, we had some evidence of missing genes. So, for example, the flowering locus T, so FT gene, was completely missing in the 12x and 12xv2 version. And now with the V4, it's back. So now we are reassured that grapevine is really a flowering plant. That's only for the joke. But if we go back to metrics, really, and this is the BUSCO to compare the V4 to the 12xv2. And what we can see is that based on the genome assembly, we were able to retrieve more BUSCO genes in the new version compared to the previous one, and they are also less fragmented compared to the previous one. So, which testified that this V4 version is improved compared to the previous one. So I told you that the genome is a mixture of Sanger quality region and PacBio regions. And so we wanted to check on that. So globally, what you have to remember is that the accuracy is very high, okay? So it's not full of errors. It's about 99.97%, so which is very high. Nevertheless, we wanted to check where the errors were. And so we plotted them on the 19 chromosomes. And we wanted to colocalize this error with where we knew where the PacBio regions were. And actually, they colocalize quite well. So you see the errors are here in red along the chromosomes, and the new PacBio regions are in blue. And basically, where we have the denser, the higher error rate, it's likely to be, we are likely to be in a PacBio region. So this is consistent with what I explained you earlier, because we know that PacBio reads at a high error rate, and so we expect to have some remaining errors in the newly assembled PacBio regions. So we'll have to keep that in mind while performing manual curation. What we also figured is that actually these new PacBio regions were enriched in heterozygous regions of the genome. So you probably know that even if PN40024 is a highly homozygous, it's not fully homozygous. And we estimate that about 5% of the genome is still heterozygous. And we could identify 7 remaining heterozygous regions. So one on chromosome 2, one on the chromosome 3, 4, 7, the largest, 10, the second largest, 11, and 15. And so what we figured is that everything colocalizes here, meaning that where we have a heterozygous region, this is where we actually perform to assemble a new PacBio regions, because these were missing in the previous assembly, and so these regions are enriched in errors. So let's say if you figure that you are working in a heterozygous region of the PN40024 genome, you may have more error to play with. So how are you going to visualize these heterozygous regions using the software that you're going to use the whole week? So this software is called Apollo. So Apollo is so this is a screenshot of Apollo, the Apollo of PN40024.v4. So we will introduce you to this tool in detail during the training school, but briefly, here you can select a region where you want to work on. So for example, here chromosome 7, and between these two coordinates on the chromosome, you have here a representation of the whole chromosome 7 pseudomolecule, and you can display different information and what we call tracks to help you with manual curation. So this you will be able to annotate the pseudomolecules along with the allelic version of the 7 heterozygous regions. So for example, here, chromosome 2 alt hetero is the allelic version of the heterozygous region that is located on chromosome 2. So that's where you could actually, you will be able to choose where you'd like to work on the manual curation. So the key points you have to keep in mind to perform manual curation, based on what I explained to you about the genome assembly. So basically, this genome assembly is a mixture between Sanger quality scaffolds and PacBio scaffolds with more error prone than the Sanger ones. But as I told you, you know that we also corrected some N regions in the Sanger scaffolds. So even previous Sanger scaffolds could have been slightly changed. So they are not completely identical. So that's why it would have been difficult to basically just transfer annotation from the 12x v2 to the new v4 version, because they are not exactly 100% identical. I told you that the PacBio heterozygous regions are more error prone than the Sanger homozygous ones. You have to keep that in mind. And also what's important is that the pattern of errors for the PacBio reads is mostly enriched in indels, so insertion and deletion. So it means that you can find a lot of times just one nucleotide that has been deleted or one nucleotide that has been inserted, and maybe wrongly. So the errors are not like Illumina patterns, which are mostly SNPs, but PacBio, these are mostly indels. So to help you, to guide you while you will be doing the manual curation with all these problems, you'll have to select here on the right of the Apollo window some tracks that we designed to help everybody with these problems. So for example, we will have access to a track showing the SNPs of PN40024 Illumina reads against itself, against the v4 assembly. And so if there are some differences between the Illumina reads and the assembly, Illumina reads are very accurate, so these may be the location of errors in the assembly and or also highlight heterozygous region. To help you locate the scaffolds, the remaining end gaps, and also the heterozygous regions, we made some dedicated tracks for that. And so, for example, here you can see the track of the heterozygous region. So when you have a yellow box like this, it means that you are actually in heterozygous region. The scaffold track here shows that we are at the junction of two different scaffolds, and the gray bars here show the regions where we still have some N in the sequence. And for the SNPs, all the green tiny bars here are differences between Illumina reads and the V4 assembly. So this helps you to figure out if you are in an accurate region or not, and it will help with your judgment when you have to perform manual curation. Another reason why we need to perform manual curation is that what's available so far is an automatic gene prediction. And we know that automatic gene predictions are only models, and that originated from a software which made a consensus of all different kinds of evidences. So these gene predictions are models, and we need sometimes some human eyes, human expertise to validate and to curate predictions. So I'm going to explain here how the PN40024 .v4.1, so this is the name of the actual automatic annotation of the V4 genome assembly, and explain you why we decided to make it de novo and fully automatic. So as I've just explained, so even the Sanger sequences between 12x V2 and V4 are not 100% identical. So even transferring the previous annotations from the previous version to the new one would have been difficult. But also what we had to keep in mind is that we needed also to annotate the new PacBio regions, which were completely missing from the previous version. So we decided not to perform a mixture of different levels of annotation and prediction. We came up with a de novo, a fully de novo, and automatic gene predictions for the V4 genome assembly. Also, the previous annotation, which name was VCOST V3, was not fully satisfactory. The genomic experts were not very keen on using it because it was too rich somehow in protein coding genes, and it counted about 42,000 predictions, which was supposed to be too high for a genome of the size and of the grapevine genome. But we didn't decide to trash the whole VCOST V3 annotation, but rather to use it as a piece of evidence to perform this new annotation for the V4. So really the goal here was to have an improved and homogeneous annotation for this whole new V4 assembly Sanger regions, but also PacBio regions. So the annotation pipeline was designed by Nicola Vitullo and Bianca Frommer in the frame of the INTEGRAPE cost action. They decided to base their pipeline on the software called Evidence Modeler, which uses all kinds of different pieces of evidence to feed the prediction model. So as I told you, we transferred the vCost v3 annotation version onto the v4, and we use that as an evidence to feed Evidence Modeler. They used protein data that were aligned onto the genome, RNA-Seq that was stranded or unstranded. They assembled transcriptome, and they used all these biological evidences to feed the model of Evidence Modeler. They also use 4 different ab initio prediction software. So ab initio, it means that they are based mostly on gene structures, so the definitions I gave you earlier on. And they decided to use several and to feed the Evidence Modeler software. So this software, we have to give some weight to the different pieces of evidence, and it computes a prediction. Then there was a filtering step, and they ended up with raw gene models. They used the software PASA to annotate the UTRs and the alternative splicing, so the isoforms. They also used the software to annotate the transfer RNAs, and we also performed functional annotation, and this at the end gave the PN40024 v4.1 gene annotation. So this is how the whole thing works basically. So the results of these gene predictions are shown and summarized in this table. So for the v4.1 annotation version, we ended up with 35,197 for the encoding genes. We found 1,809 protein coding genes in the alt region, you know, it's the allelic version of the heterozygous regions, and some tRNAs as well. So you have all the metrics here. And what was very interesting is that we were able to annotate 1,572 novel protein coding genes located in the PacBio regions. So this, which represents 4.5% of the total number of genes. So these genes were completely missing in the previous genome assembly and annotation, of course. And so these numbers are very nice because this is what we expect. So about 35,000 protein coding genes for grapevine genome, and not as much as what we had previously with the vCost v3 version. So we were quite happy with these numbers, but we also needed to test whether the genes were accurate and true. So to do that, we run some BUSCO on the gene predictions, and here we compare the v4.1 annotation, the initial prediction with the the vCost v3 annotation prediction, and the new one performs better than the previous one with less missing genes, less fragmented genes. And so this, we are pretty confident that this new annotation was much better than the vCost v3. So you'll have to still the VCOST v3 version could be quite informative while you will be performing manual curation. But as I told you, and you have to keep that in mind, even if the Sanger regions, the two versions of annotation may not be identical because the genome may not be identical in these regions. So to check whether the new v4.1 prediction performs better than the previous one, we designed for you some tracks on the Apollo. So the track for the v3 annotations that were transferred to the v4 genome assembly, and also the previous one, which name was v1, which was noticeably good. And so usually the grapevine community used to like this one better actually than the v3. And so to really keep track of these two annotations, we designed some tracks that we can visualize on the Apollo to help for manual curation. So the third reason to perform manual curation is that we need to double check and to validate genes that are important for the grapevine community, for which we already have expertise and functional information about. And so it's also important to ensure traceability between the version that we have in hands. So to do this, and so this, I'm going to introduce you to the great work that the group of Thomas Matheus performed last year. So they checked, they looked at all the grapevine literature to find some the genes for which the community has expertise and has a validated function. So at different validation levels, but still for which we have information about the function. And so we figured that these genes that are now listed in what we call the gene catalog for the grapevine community. So these are really the most relevant, the most important genes for the community because they've been worked on extensively. And so we figured that this gene, at least this gene catalog, needs to be nicely transferred from the previous annotations to the v4.1. And so that's why we need to perform manual curation, especially for these very important genes to the grapevine community. So there is a special effort dedicated to these genes. So this is really a matter of helping the community to track back their important genes from previous annotation to this new one. And so this gene catalog is of special interest when we perform manual curation, and that's why we design a dedicated track for the gene catalog, so which is actually transferring the sequence that we had from previous annotation to the v4.1. And so this helps to accurately manually curate these very important genes. And so we'll probably focus during training school our efforts on this gene catalog, and this is where the efforts were put a lot already during the last year. But it's not only about these very important genes, but it's also very important in general to keep traceability between different versions, so that everyone who's would like to use this new version will not be completely lost, because the gene names would be different and everything would be different, and they have to start from scratch. So we didn't want that. So we wanted this new genome assembly and this new genome annotation, gene annotation, to be as useful for the community as possible. So we made a huge effort into transferring gene names from the VCOS v3 to the v4.1. To do that, we started with reciprocal best hits between the two annotations. And for the pairs of genes that had a reciprocal best hit between the two versions, we performed filtering. There needed to be 70% coverage and identity between the two genes. They needed to be on the same chromosome, and overall the collinearity, so the neighbor genes should also have the conserved name. And so we checked also collinearity to testify that we could keep the same name from VCOS v3 for this v4.1. So overall, we were able to transfer, so to reuse previous gene names for 76% of the genes, so more than 23,000 gene predictions. So they have the same ID, same name in the v4.1 than in the VCOS v3. However, there are about 12,000 gene predictions for which we had to give a new name. So it's about 34% of the gene predictions. And to distinguish between these genes with new names and the one that kept the old names, we decided to name the genes with new names, starting with 4 here. So just to remind you the way the naming system in grapevine. So it starts with a VITV for Vitis vinifera, then 2 digits for the chromosome number, then a G for gene, 0 always, and then the 4 unique digits for the being the name of the gene. And so, as I told you, when we have a name that has never been given before, except now in the v4.1, it starts with the 4. Yeah, so 4,000 whatever. So for the v4.1 nomenclature, that's how it is. For genes for which we could transfer the gene name, so it remains the same. And we also keep track of all the previous names that were given in previous annotations. So it helps researchers and scientists to keep track of their favorite gene. So as I told you, the novel genes, so they start with the 4 here. The genes that are annotated in the alt region, so the allelic version of the heterozygous region, they have an underscore alt at the end with the name of the allelic version on the molecule. And then we have this nomenclature, very typical for transcripts, coding sequence, exons, and UTRs. So the key points here to remember is that when you have a name that is kept from the VCOS v3 and transferred to the v4.1, so the genes may be very similar, okay? So the predictions may be very similar, but not always identical. So remember, the filtering was not 100% identity, 100% coverage, but we allowed some differences. So it guarantees that it's the most similar, but not completely identical. On the other hand, the new genes, so starting with the 4 here, are supposed to be different to anything annotated previously in the VCOS V3. You may end up with some partial similarities, but not on the full coverage, for example, of the gene. But anyway, for manual curation, so this is just for you to understand how we did that and how it works. But you won't have to think of, is it a gene name that I need to transfer or not? Do I have to change the name? No, don't bother about that. This is going to be handled automatically by a script later on when we are going to integrate the manual curation into the second or the third version of annotation. The only thing you should be worried about if you are working on a gene part of the gene catalog, we need to keep track of the name and most of all the symbol of the gene. And that's where you will have to keep track of it. But we'll go back to that during the training school. But so yes, there are some specificities if you are working with genes from the gene catalog. So the fourth reason why we need to perform manual curation is that especially when we are working on genes that are part of large gene families. So for example, and especially when the genes are clustered, it means that for example, here I took a screenshot of the stilbene synthase gene family. So here on this part of chromosome 16, there are more than 42 stilbene synthase encoding genes that are just neighbor and next to each other. And so they are so similar that usually the prediction softwares, they just messed up. So it's very, very difficult even for the softwares to annotate the single gene copies individually. So for example, here I have a cluster of 6 stilbene synthase genes, and the V4.1, which is this track here, was only able to annotate 1 here, which looks correct, but then it missed this one, this one, this one, this one, and also that one, and that doesn't look very accurate. So let's say even our best annotation pipeline didn't perform well at all for a clustered gene family like stilbene synthase gene family. So this is where especially software cannot replace human expertise, and we really need to go into all the available knowledge that we have to distinguish all these gene copies. And so this means, and we will go back to that during the training school, this means that we could use all other resources than the Apollo. So for example, we will introduce you to the gene tree. You will have to use probably some online blast at NCBI or InterProScan to look for the specific domains that are important for a given gene family, and in order to be close as possible to the truth. So this is a very important point I'd like to make, is that structural prediction, I mean gene predictions or even manual curation, it's supposed to be a model of the truth, means that we'd like to be the closest possible to the truth, but sometimes it's very difficult, and you will face some situation like that probably, to make a clear-cut decision. But you'll have to. So you use all the evidence, you will use all the evidences that you have in hand and make a decision. But if you ask your neighbor, maybe he or she is going to make a different decision compared to yours. But it would be almost impossible to know which one is the closest to the truth. So an annotation, a manual curation, we know it's never perfect. It's just the best we can perform, the best we can do. So this is a case, especially with clustered gene families, where sometimes we have to make some decisions, but we are not very, very 100% sure that it's the truth. But you'll perform your best, and that's all we ask for you. And then the very last reason why it would be useful to perform manual curation is that actually we have some new PN40024 that are available. So these data are RNA-Seq data from the PN40024 genotype itself. And this is brand new, and it was not used by the automatic annotation pipeline. So it was not a piece of evidence that was used to feed the pipeline. The transcriptome, the RNA-Seq data sets that were used came from other varieties, other genotypes, but not PN40024. So this data may not give as accurate results as using the PN40000 RNA-Seq data set itself. So probably the use of this track, so we designed this track, so the PN40024 RNA-Seq 2022, will help to make decision. And this is the most accurate data set possible. So just to give you a little bit of detail here, this track is the result of the merging of alignments from different RNA-Seq samples of PN40024. So we sequenced more than 10 different biological samples of different organs, different developmental stages of the PN genotype. We got that, so extracted the RNAs, here the poly A, everything got sequenced, aligned against the PN before genome assembly, and all of that was merged together so that we have only one single track to look at and to help to decipher transcription start site, but also exon intron borders, and also the contiguity of the gene annotation. So this is going to be a very, very useful track for manual curation. So at the end of the manual curation, we will have improved the automatic prediction a lot. So how do we perform manual curation? So what are the tools that you'll have in hand and that we are going to teach you how to use? So this Apollo, of course, that I presented you already, which is an editable JBrowse. So JBrowse is a genome navigator, an interface where you can explore a genome. But this, the specificity of an Apollo is that you have this yellow card here, where you can make some edits, and it's impossible if you are only using a regular JBrowse. So it allows, Apollo allows you to make some edits, and this is what we need. So the Apollo we are going to use is available with this link, and you'll have to use a login and password that we're going to provide you during the training school. You could also use the sequence server, so which is a homemade BLAST server that we made available for the community. So you can blast any sequence that you get from the Apollo to check it against the 12x2, the v4.1 annotation, but also the assembly, the protein sequence. So this is also very useful when you have to double check your work. You will have access to the Grameen gene tree, which is a tool that was designed and developed by Doreen Ware's group at the Cold Spring Harbor. So this allows to compare multiple gene predictions from different grapevine varieties. So the link to access it is this one. And so this is the tree that you can get for this gene. But if you want the gene tree for another gene, you'll have to change that in the link. And so this helps to compare. So the annotation, the v4.1 prediction that is up there, to see if it has a similar structure in terms of length, in terms of exon, etc., compared to all the other ones. But you're going to be introduced to that tool as well during the training school. You will have to use probably online BLAST interface, for example, the NCBI one, but you can use basically any. I also like to use InterpoScan to check whether I have the functional domain that I expect for the gene I'm actually creating. So these two tools are also very useful. And last but not least, you'll have to use and carefully read the curation guidelines that the group of trainers wrote and designed, and especially David, who you're going to meet him during the training school, and he was in charge of writing these guidelines. And these guidelines are here to explain you and to detail every step of how to perform manual curation in grapevine, and all what's recommended for everybody who wants to perform manual curation using Apollo. So now to finish, I'd like to explain you what we expect from you during this training school and afterwards. So this is a list of what we expect from you. So during the training school, we expect you to check if the automatic annotation is correct using the Apollo tracks that are available. If you think it's needed, you can edit the annotation. It's not always needed. You can just validate an automatic annotation because it's correct. So this happens, and we like these situations a lot, but because it's very fast. So you may then edit if it's not completely accurate in your sense. Then if you make some edits, you need to validate your manual curation using some other tools to check whether the edits that you suggested are possible. Then if you are working on a gene that is part of the gene catalog, or let's say if you are an expert in some kind of genes, then you are allowed to give the name and or the function of the encoded protein for this gene. Then we ask you to give some information about the edits you made. So we will explain you that during the training school, of course, but this is for a matter of traceability. We'd like to know which edits you made and why, which pieces of evidence guided you through these edits. Then this is a very general point, but make sure you stick to the guidelines because we took a lot of time altogether to think of these guidelines and to give the rules so that every curator stick to the same rules, and we want to end up with something that is homogeneous, even if we had tens of different curators, which is the case already. And then at the end and later on, we expect from you to disseminate your manual curation skills, but only when you feel comfortable enough to do so. The idea here is to train you, but also that then when you feel comfortable enough, that you guys would train some other people in your lab or in the grapevine community, so that we may end up with a curated annotation, which is the largest as possible. And this will be perfect. So I'd like to end my lecture today with acknowledgements. I'd like to thank Amandine Welt and Marie Lahaye, who so Amandine performed genome assembly, and Marie helped her with putting together the Apollo, the JBrowse, the BLAST server in my group. So very many thanks to these two girls. Jérôme Grimplet, who organized and hosted the training school, but also for his help with gene nomenclature and gene transfer. The group of Thomas Matheus with David Navarro and Luis Orduna, who helped a lot with the Apollo as well, and to draft the guidelines. Nicola Vitulo, Bianca Framer, Daniela Olgrave for the annotation pipeline that we used. The group of Doreen Ware is Marcella and Camille, who were involved in developing the gene tree interface that we are going to use. And they helped us a lot to give us some feedback on how annotation was thought and performed in other plant species. So this was a very useful feedback. We'd like to thank Anne-Marie Digby, who is the person without who this training school wouldn't have been possible. She's handling all the administrative stuff and contact with the cost for reimbursement and organization. So many, many, many thanks to Anne-Marie. We miss you a lot. We will miss you a lot, Anne-Marie, in Spain. And last but not least, Mario Pezzotti for chairing the cost action. And here are some pictures from the PN40024 plant. As you see, so it's growing nicely in the greenhouse near my lab. And so these are some pictures of samples that we took for the RNA-Seq. So all of this training school was actually born from a previous workshop that we hosted here in Colmar. And so we had a great time altogether. It was 1 year ago. And that's where we thought about the guidelines and how to perform and how to help the grapevine community to achieve manual curation in the most efficient and correct way possible. So many thanks to all of this dream team. And then I'd like to thank you for your attention, and I'm really looking forward to either see you in Zaragoza or online to discuss more about manual curation and to answer your questions. Bye-bye and see you later.